All right, welcome to our second video for Chapter 16. In this video, we're looking at the lactose operon as well as epigenetic regulation of gene expression. So in our previous video, we talked about the tryptophan operon and negative regulators. Now we're going to switch gears and look at the lactose operon and positive regulators that turn genes on and activate their transcription. So one example is the lactose operon. Again, we're still looking at prokaryotes because prokaryotes, remember, their messenger RNA has multiple genes and they are organized into these structures called operons. And we're going to look at the lactose operon in bacteria like E. coli. It turns out that most cells prefer glucose. Glucose is their main energy source. But when you have little or no glucose, when their levels fall, E. coli and other bacteria have the ability to use other sugars. They can metabolize other sugars such as lactose for energy, but they need different types of enzymes to break down these other sugars. So we're going to look at the lactose operon, and in the lactose operon, we're going to see genes that are present to encode enzymes that will take in lactose into the cell and allow the processing or metabolism of lactose to produce energy. For these positive regulators, we're going to look at cyclic AMP and CAP, which stands for catabolite activator protein. It turns out that when glucose levels are low, so low glucose, when glucose levels are low, you're going to have greater levels of cyclic AMP in the cell. Cyclic AMP accumulates in the cell. This is a signaling molecule that's involved in energy metabolism. And when you have a lot of cyclic AMP, it's going to bind to catabolite activator protein. And this complex together, cyclic AMP and CAP, is going to bind to the promoter region. And what that does is it actually helps stabilize RNA polymerase so that RNA polymerase has a greater transcription rate of these genes that are going to end up being used to catabolize or break down lactose for energy. So when glucose levels are low and you have other sugars such as lactose available, you're going to want to transcribe these genes that allow lactose to enter the cell and allow the cell to metabolize lactose for energy. And how this occurs is, for example, shown down here. This is the first step. Glucose levels become limited, so glucose goes down, and cyclic AMP levels go up. What happens next, as I mentioned, is cyclic AMP binds to the CAP protein, and these are known as positive regulators. They're going to bind to that promoter region upstream of where RNA polymerase binds, and the CAP-CAMP binding to the promoter stabilizes RNA polymerase so that it binds even well or even stronger to the promoter region and increases transcription of these lactose catabolism genes. Interestingly, the lac operon or lactose operon is opposite of the tryptophan operon because it's an inducible operon. What this means is the default state of this operon is off. And this is because we usually do not break down lactose for energy. We're usually breaking down glucose for energy. That's our really primary or desired sugar source. And you only use lactose when you have to. So in the absence of lactose, which is more common, the common state, the repressor protein is bound to the operator region and physically blocks RNA polymerase. And this is because for this repressor, the repressor of the lac operon, the shape of the protein is naturally such that it naturally binds to the operator. It's optimized to bind to the operator region and blocks RNA polymerase from transcribing these genes. Oh, it's only when you have lactose, so in the presence of lactose, that lactose will bind to the repressor and change its shape so that it no longer binds to the operator. And now RNA polymerase is free to transcribe the genes that will allow the intake and breakdown of lactose for energy. So this is called an inducer. Lactose is the inducer because it binds to the repressor protein and makes it stop binding to the operator, allowing transcription of the genes. This is an inducible operon because of this. And again, this really depends on the needs of the cell, much like the tryptophan operon. When you have lactose, then you want to transcribe the genes that allow the enzymes to metabolize the breakdown of lactose. Notice, however, that if you just have lactose available and you don't know anything about whether glucose levels are high or low, I don't see that cyclic AMP or CAP here. This means that transcription will occur, but it goes at a slow rate. It's only when you have a high level of that CAP-CAMP complex that transcription will occur at a greater rate. And we see that scenario in this upper picture where lactose is available. And I can tell that lactose is present at the moment because the repressor protein is not bound to the operator. So RNA polymerase is free to move forward and transcribe these genes. This picture indicates the presence of CAMP and CAP, which tells me that there's also low or no glucose currently available. So in the presence of lactose and little or no glucose, the transcription of the mRNA for these lactose metabolism genes will increase. So in contrast to the low transcription rate we saw in the previous slide, here we have a greater transcription rate. Notice though, down here, even though we have CAP and CAMP, if there is no lactose, there will still be no transcription. So if there's no lactose, but also little glucose, which is indicated by the cyclic AMP and CAP complex, there's still no transcription. So you have to have both lactose present and the CAP-CAMP if you want high rates of messenger RNA transcription of these lactose metabolism genes. And underneath this figure in our textbook, they pose the question, in E. coli, the tryptophan operon is on by default, while the lac operon is off. Why do you think this is the case? So if we go back and think about the two operons, remember that the tryptophan operon, the purpose is to make tryptophan, and tryptophan is an amino acid that's essential for proteins. So we always need to have some tryptophan in the cell. If there's too much tryptophan, however, it's wasteful to make more, so we will repress the tryp operon at that point. In contrast, lactose is a sugar that's not always available to cells. So it does not make sense to make the enzymes necessary to digest lactose unless it's available. So the lac operon is only on when you have lactose that's present in the cell. And our book provides a table that tells us the conditions where you will be transcribing the lac operon. So for this last column, you can think of it as, is the operon on, yes or no? So if I look at the first row, if glucose is available, but CAP does not bind and there is no lactose, that means the repressor will be bound to the operator and you're not going to have transcription of the lactose operon genes. So that makes sense. If there's no lactose, why would I make enzymes to metabolize lactose? All right, let's look at the second row. I have glucose available, so that means there's no cyclic AMP, so CAP does not bind. I do have lactose available. When lactose is available, it binds to the repressor protein, so it doesn't bind to the operator. So that means RNA polymerase is free to move forward and transcribe the genes. So there's some. Remember, there's a low rate of transcription because there's no CAP protein with a cyclic AMP bound to the promoter. In the next case, we have no glucose. So the CAP-CAMP binds, but there's no lactose, If there's no lactose, again, it would not make sense to make the enzymes to break down lactose. And then let's look at the last one. There's no glucose. There is CAP bound to the promoter site. There is lactose. So this is when we have the highest rate of transcription of the operon, of those lactose catabolism genes. And I didn't talk about this because these were similar to the above, but remember that whenever lactose is available, then the repressor would not be able to bind to the operator. When lactose is not available, then the repressor will be bound to the operator to prevent RNA polymerase from moving forward. So for the operon so far, we've been talking about prokaryotes. Remember, operons are present in prokaryotic cells like bacteria. And now we're going to switch gears. Let's talk about eukaryotes. Eukaryotes, and we're going to talk about gene regulation starting with epigenetics, epigenetic gene regulation. Remember that eukaryotic gene expression is more complex because we have that nucleus. So transcription happens in the nucleus, translation is going to happen in the cytoplasm. And remember, regulation happens at many different levels rather than just the transcriptional level that we saw in prokaryotes. So the very first level of control really starts with accessing the DNA. Are you even able to open up the DNA to read it and make messenger RNA? That's the epigenetic regulation process. And it occurs even before you can start transcription. So there are two subcategories to epigenetic regulation. One is chromatin remodeling, and this has to do with how DNA is associated with those histone proteins. Is it tightly bound to histone proteins, or is the DNA loosely wound around the histone proteins? The second one, these are not the only two, but the only two that we really talk about so far in this chapter. The second one is DNA methylation. And this is really associated with developmental changes that can occur as you age, as well as gene silencing. And we're going to see that if you methylate our DNA histone complex, you're going to end up usually silencing genes. So this usually is silencing like we see here. Later on in this chapter, we're going to see that transcription factors are also important. These are proteins that are going to regulate transcription of our genetic information. If we look at how the human genome is regulated, we can kind of see why epigenetics is so important to gene expression and the regulation of gene expression. So our human genome encodes over 20,000 genes across those 46 chromosomes and 23 pairs of chromosomes. So we have 23, I should say really pairs of chromosomes because we have 23 from mom and 23 from dad. And within each chromosome, there are thousands of genes. We know from previous chapters that DNA is wound around histone proteins to compact it into chromatin. And if we want to express these genes, remember that means we were going to read DNA and make our messenger RNA, then we have to unwind, kind of loosen the way DNA is wrapped around those histone proteins so it's available to the polymerases. And specifically, remember, these are RNA polymerases that read DNA and make messenger RNA. So it's very organized. DNA, the way it's organized in the nucleus, has to be in a way that it's accessible, or the genes are accessible when they're needed by specific cell types. We've seen the organization of DNA around histone proteins several times already. Remember that this structure that looks like beads on a string is a nucleosome, and under the electron microscope, you can see tiny little circles everywhere, and these are our nucleosomes, these beads on a string. DNA, we remember, is negatively charged overall because of the phosphate groups, and then histone proteins are positively charged, so there's usually a pretty strong interaction between the two. If we think about adding methyl groups or acetyl groups, we'll see later on as well, these change the interaction between DNA and histone proteins to either tighten the interaction or loosen the interaction. And in general, methylation of these complexes will result in gene silencing. And then adding acetyl groups to these complexes will really result in gene expression, or greater gene expression, I should say. And we see that here, how nucleosomes are controlling the access to DNA. So when I say access, this is really access from or by the RNA polymerases, which need to access DNA in order to make messenger RNA. oops, sorry, right there. And I see the methylation over here. Methylation usually causes the packing tightly, causes the nucleosomes to pack tightly together. You can see there's no space, and if I want to access this gene right here, it's not accessible. So this means that genes are not expressed when the DNA and histone complexes are methylated. So gene expression is turned off. And this is the general rule when it comes to methylation. Not always true in all species and all cell types, but for our course, we're going to say methylation silences genes again. At the bottom picture, I see histone acetylation. Acetylation results in loose packing of those nucleosomes. You can see so much more space between the nucleosomes, and here is my gene of interest. Now it's accessible, and I can express these genes. So when nucleosomes are far apart due to the addition of these acetyl groups, histone acetylation, we're going to allow gene expression to occur. So that will be our general rule, that acetylation increases gene expression, whereas methylation silences these genes. So we call these additions, these methyl or acetyl groups, we call these chemical tags that are added to histones in DNA, and they're not permanent. They can be added or removed depending on the environment, the diet, and other factors. These really act as signals to tell our histones if the regions of the chromosomes should be accessible or inaccessible. So again, this is known as epigenetic regulation, which means above or around genetics. These are temporary changes, not always temporary. Some of them can be quite long-lasting, but they do not alter our nucleotide sequence. They affect gene expression by, again, determining whether or not those genes are accessible to our RNA polymerases. And how some of them work, as I mentioned earlier, for example, acetyl groups will impact the charge, the usual charge of DNA, which is negatively charged, binding to histone proteins, which are positively charged. If you add things like acetyl groups, the charge of the histone proteins becomes less positive than usual, allowing that DNA to relax more around those histone proteins. So we saw earlier that our nucleosomes can be methylated, or other chemical tags can be added to loosen or tighten the wrapping of DNA around those histones and production of nucleosomes. So that was here. In fact, DNA methylation can also occur directly. So if the DNA itself is methylated, this usually happens in specific regions we call CPG islands. These are called CPG islands because in these regions of DNA we have a high frequency of our cytosine and guanine nucleotides. And these are in the promoter region of our genes. These are upstream of the gene of interest. Just like adding methyl tags to the nucleosomes, methylated genes are also usually silenced. And in fact, genes that are silenced during the development of gametes, this is from a parent or the other, or both, this silencing can be transmitted to the offspring. And if this happens, these are known as imprinted genes. So what happens during your lifetime due to diet or due other environmental stressors can cause your genes to be methylated or silenced, and you can pass that on to your offspring if this is happening in gametes. So again, many different things can affect methylation patterns: your diet, the environmental conditions, other stressors, like those that we saw in the article and the film that we watched recently. So we've seen epigenetic changes to chromatin can result from things like development. It can happen while you're still in the womb or during childhood, due to exposure to chemicals or different drugs, due to aging, or even your diet. And these changes can result in a variety of effects: cancer, autoimmune disease, mental disorders, diabetes, and even more. Why does it matter if we have access to our DNA, to our genes? Remember that in order to read DNA and make messenger RNA, we need transcription factors to bind to the promoter region and other upstream regions that enhance transcription and initiate transcription in the first place. And this can only happen if our DNA is accessible. Twin studies are big in many biological fields, but also in epigenetics. And a few years ago, NASA published a study regarding these twin astronauts, where Scott Kelly was the twin that went into space, whereas his brother stayed here. And what they saw was that in Scott Kelly's genes, there were so many changes while he was out there, and those changes remained for a few months after he came back to Earth. His telomeres got longer in space before they shrank back to normal, or actually sometimes they even got shorter after he returned to Earth. They also saw methylation changes compared to his twin, and luckily most of the changes returned to normal. There are some changes that have not returned to normal, but we're not sure if they're just different because of aging and time, or if it was because of the mission. And that takes us to the end of our second video. In our third video, we're going to look at eukaryotic transcription and gene regulation and the role of transcription machinery in gene expression.